Right. Hello, everyone. Now I just want to talk a little bit about uh, when there is an error in DNA replication or when an external stressor or environmental factor is going to damage DNA in some way and cause a mutation as a result. A mutation simply being, we haven't talked too much about them just yet, but it's simply being a change in the nucleotide sequence on one of the two strands, which would actually result in a change in both strands in the next round of cell division, which, is just a nucleotide base pair that's being changed as a result, right? So what causes this? What are the implications? That's what I kind of want to talk a little bit about. And so let's start here. Okay. And specifically, I'd like to focus on repair and the repair mechanisms that the cell employs to fix these DNA errors or DNA damages that can occur. Because they're very important in the fact that we have evolved these mechanisms to prevent cancer, to prevent genetic diseases and really, harmful effects to the cell and to the organs and to the organism as a whole. And so I thought this was kind of an interesting fact to start with. So on an average rate of about, during DNA replication, as we've seen, DNA polymerase 3 specifically adds complementary nucleotides, transfers them in as the helicase unwinds the double helix and you proceed down the original parent molecule, and DNA polymerase is transferring in millions and millions of nucleotides, billions in all the different DNA molecules, really. In the human genome, we have roughly 3 billion total nucleotides. So, but if you take a look at this, the DNA polymerase 3 is transferring in complementary DNA nucleotides, right? And so what does that mean? It means if there's a T, it's going to add an A to that new strand. If it's a it would add a T. If it's a G, C, et cetera, et cetera. Well, on a rate of about 1 in 10 to the 5th, which is, what is that, 100,000, right? 1 in 100,000, it puts in the wrong nucleotide. So like if there's a T, it puts a C there. Or if there's a G, it puts an A or something, right? On accident. I don't blame it. It's doing millions in seconds. Like the fact that it's still that small is pretty remarkable. But then the completed molecule, any of these types of errors, there's roughly 1 in 10 billion, 10 to the 10th, nucleotides, a rate that's 100,000 times lower. So how is it correcting these mistakes? Or how does the cell correct these mistakes? And I'm sure you won't be surprised to know that there are enzymes and enzyme systems involved. But even in addition to that, the DNA polymerases themselves have this ability within, or an intrinsic quality, that if they accidentally put in the wrong nucleotide, they can quickly change it out, put in the right one, and keep going. So there's this intrinsic quality of proofreading that DNA polymerases have. But even that's not enough. And sometimes it still will keep going. And so we have backup plans on backup plans, right? Because again, we evolved this to prevent things like cancer, and to, again, mutations are almost always, almost always really harmful to the cell. And that functional protein product can't be produced from that DNA, from the gene, if you have a harmful mutation there. Which we're going to talk a lot more about that process when we talk about central dogma. But the point is, we need the correct DNA sequence, and we need accurate DNA replication. And so here's one backup plan: mismatch repair. So sometimes the nucleotides evade proofreading, in which case MMR, or mismatch repair enzymes, can come in and assist and take out that incorrect nucleotide and put the right one in. Classic example, now this is kind of wordy, but it's all on D2L for you. And it's just phrased really well, so I wanted to keep it wordy. But essentially, if there's damage or replication errors, a classic example of a mismatch repair system is nucleotide excision repair. And so there's a type of enzyme called a nuclease, and a nuclease will cut DNA. So it actually will cut straight through the double helix. In a lot of cases, it can actually cut through both strands. It depends, but point being, it's going to cut the DNA and remove the incorrect nucleotide, or nucleotides if it's a mutation of, if there's incorrect more than one, sometimes there's 2 or 3. And then it'll actually fill in the gap that's exposed with the correct complementary DNA nucleotides. Of course, that's separate from the nuclease. It's a DNA polymerase will complete the job, as well as DNA ligase that we've talked about is involved in connecting the sugar phosphate backbone of the DNA double strand, the strands of DNA, the sugars and the phosphates of those adjacent nucleotides, okay? And again, the classic example is nucleotide excision repair. Here's a cool image of this. You've got this incorrect, it looks like more like a thymine dimer. This is blocking my view, but sometimes ionizing radiation, ultraviolet radiation from the sun can cause these little kinks, these thymine dimers. But it's just damage to DNA that makes that can lead to incorrect nucleotide pairing. Point being is we need to fix it. And so this nuclease enzyme will come in and cut the strand of DNA at that location. DNA polymerase will then come in, fill in with the proper complementary nucleotides. And last, DNA ligase will connect the final nucleotide here to the undamaged one, just by forming a bond between the two, ligating it and sealing it off, I guess. So yeah, and I guess the last thing I can mention here is mutations, and this DNA alteration can happen from, as we would say, spontaneously, naturally occurring from errors in DNA replication. Or there can be an external cause of DNA damage. And of course, DNA molecules are constantly subjected to potentially harmful chemical and physical agents. And I would, carcinogenic compounds in food or tobacco smoke, even secondhand smoke. But radiation is also a really common physical agent, X-rays, ultraviolet radiation, that can cause DNA damage and would, induce the application of a DNA repair system such as this. Okay, well, that's it for DNA repair, enzymes, and that kind of mechanism. And I'll see you next time.